The replication of DNA first begins with the helicase enzyme. Helicase naturally works to unzip the DNA bases. However, to do this, gyrase must help helicase by untwisting the DNA. As soon as the DNA strands are finally separated and untwisted, the single-stranded binding proteins, also known as SSBP, you heard right, bind to the DNA backbone and ensure that the strands stay apart. After this, the enzyme primase synthesizes and adds short RNA sequences called RNA primers to the strands in order to construct the RNA landing zones. With this, DNA synthesis can finally begin. An enzyme called DNA polymerase III lands onto the RNA landing zones and begins to read the strand from the 3' end to the 5' end, while also writing on a new DNA strand from 5' to 3', so on one strand known as the leading strand. The DNA polymerase III will follow the helicase enzymes. Therefore, the DNA polymerase III will continuously synthesize new DNA nucleotides on this strand. In contrast, on the other strand, the DNA polymerase III will synthesize DNA in the opposite direction that helicase is moving. On this strand, called the lagging strand, the DNA is synthesized in fragments. These fragments are called Okazaki fragments. The enzyme DNA polymerase I will then replace the RNA primers with DNA nucleotides. Each Okazaki fragment cannot connect to each other naturally, so an enzyme called ligase must connect each of the fragments by forming phosphodiester bonds between them.